Hello everyone, I am here to share you some points regarding the pheochromocytoma. Let's begin with what pheochromocytoma is and what are the various molecular pathogenesis of this pheochromocytoma, and we also learn about the morphologic features like the gross features as well as histologic features in this part. So let's begin with the introduction. Actually, this pheochromocytoma is the neoplasms that is composed of chromophil cells and that synthesize and release catecholamines. And there is the traditional rule of 10 years in pheochromocytoma that represents what features could be seen in pheochromocytoma with the relation to the number 10. First, there is the traditional belief that 10% of the pheochromocytoma are extra-adrenal. That means this catecholamine secreting neoplasm can be present at the site other than this adrenal gland, and that could be any site like organ of Zucker candle. That means the cluster of the chromaffin cells present near the abdominal aorta. Next could be the carotid body, and other could be like sympathetic and parasympathetic nervous system, which is referred as paragangliomas. In the picture, we can see the various parasympathetic and sympathetic trunks are being affected, and the neoplasm present at such site to produce catecholamines similar to um, the adrenal pheochromocytoma that results in the excessive synthesis as well as release of the catecholamines. Next, we see that 10% sporadic are bilateral. That means 10% of the sporadic pheochromocytoma are bilateral. That means that are present in the both of the adrenal medulla of the both of the suprarenal gland, and this may rise to as high as 50% cases when associated with the familial tumor syndromes. If this pheochromocytoma is associated or it comes in association with any other familial tumor syndromes, then at that case this 10% occurrence may reach up to 50%. Next is 10% adrenal pheochromocytoma are biologically malignant. However, malignancy is more common in 20% to 40% of the extraadrenal paragangliomas with certain germline mutations. And various modification can be seen. That means 25% have an oncogenic germline mutation, and the 10% adrenal pheochromocytoma may not be associated with the hypertension, but about 90% of the patient may present with the hypertension, and this is a dominant feature of the pheochromocytoma. And out of these 90% patient with the hypertension, two-thirds may have paroxysmal episodes with a sudden rise in the blood pressure and the palpitation, and that can be very fatal. And patients with the germline mutations like the bilateral tumors are typically younger than the sporadic tumors. That means sporadic type of pheochromocytoma occurs in the young aged people. Next, we see the pathogenesis of the pheochromocytoma. Generally, the gene affected in the pheochromocytoma fall into the two classes. First is that those that enhance the growth factor receptor pathway signaling, for example, like the RUT and NF1 gene. Such genes enhance the growth factor, and the growth factor is a very much vital thing for the growth of any cell, differentiation as well as proliferation of the cells. So one category of the gene affected gene fall under such category. Next type of the genes fall into the those that increases the activity as well as stability of the hypoxia-induced factor 1 alpha, hypoxia-induced factor 2 alpha. This is the second category where the affected gene directly um, affect the activity as well as stability or degradation of these hypoxia-induced factors, HIF. And this create the pseudo-hypoxia phenotype that we see in the later slide. Where BHL syndrome is associated with many tumors, and this can be taken as an example of the familial tumor syndrome, where the pheochromocytoma and paragangliomas are very uh, common to occur in case of the VHL syndrome. And VHL syndrome, there is a disruption of the VHL genes by loss of function mutation in the VHL gene. Normally, this VHL gene encode tumor suppressor proteins, and that suppressor proteins are required for the oxygen-dependent degradation of this hypoxia-induced factor 1 alpha. And if there is a mutation, a loss of function mutation in these VHL genes, then those proteins couldn't be um, synthesized. So the degradation of this hypoxia-induced factor 1 alpha will be compromised. And next we see that polycythemia paraganglioma syndrome, where there is a gain of function mutation in EPAS1 genes, and this mutation occurs as the amino acid residues which is required for this hypoxia-induced factor 2 alpha degradation. So mutation in such genes are at this amino acid residues results in the compromised degradation of this hypoxia-induced factor 2 alpha. So APS1 gene normally encodes for this HIF2 alpha, and in pheochromocytoma there is also germline mutation of the succinate dehydrogenase complex, and this most commonly occur in the succinate dehydrogenase complex B, C, and D. And this mutation lead to the upregulation of the hypoxia-induced factor proteins. And also we can see the germline mutation involved in the sporadic chromocytoma as NF1, VHL, EPS1, and RAT. Such germline mutation is very common in the sporadic pheochromocytoma. So these are the pathogenesis of pheochromocytoma. And first here we see the normal function of hypoxia-induced factor 1 alpha and 2 alpha in our body. How is it beneficial to our body, and why is it necessary to our body? And, generally, these are the transcription factors that since the hypoxic condition developed inside our tissues and organs, and after sensing those, this hypoxia-induced factor 1 alpha leads to the metabolic shift from the oxidative phosphorylation to the glycolysis, whereas this 2 alpha senses chronic hypoxia. And after sensing this hypoxic condition, they both involve in angiogenesis, and their main target will be to supply adequate amount of oxygen to the compromised tissues or the tissues undergoing ischemia. So these are crucial for the tissues to prevent from ischemia and the necrosis. But in case of pheochromocytoma, there is a mutation in VHL and SDH and any other genes that disrupt this normal degradation of the hypoxia-induced factors. These factors are necessary once the development of hypoxic condition in the body, but once this condition is recovered, then this hypoxia-induced factor need to be degraded. But the mutation in such gene like VHL, SDH, and any other genes result in the disruption of the normal degradation of such transcription factors. And what does it do? It leads to a constant hypoxia-like signal. Even the oxygen level inside the tissues and organs are normal. So this creates pseudo-hypoxic state in the adrenal medulla, and this triggers the hypoxia-induced factors uh, to induce uncontrolled proliferation and differentiation of the chromophylls, resulting in the excess secretion of these catecholamines. So such things happen in the pheochromocytoma. So then now we are going to morphology. First we see grass. Here we can see that this in pheochromocytoma, the adrenal medulla is mainly affected, and it could range from small to large hemorrhagic masses, and they have well circumscribed lesions confined to the adrenal, and some extra adrenal, you know, pheochromocytoma may also present, and there's another thing. But in adrenal medulla, the lesion is circumscribed, and that ranges from small to large hemorrhagic masses, and their average weight is 100 gram, but it may range from 1 gram to 4,000 gram. So large tumors is demarcated by either connective tissues or that is compressed cortical or medullary tissues, while the lobular pattern-like appearance can be appreciated in pheochromocytoma because of the presence of the richly vascularized fibrous trabeculae. The trabeculae of the adrenal medulla will be richly vascularized because we studied earlier that hypoxia-induced factor enhance the angiogenesis to supply more amount of the oxygen to the tissues. And here, even though there is no hypoxic condition, but the um, hypoxia induced factors are present and that are doing their function, that is angiogenesis. So the trabeculae are richly vascularized due to the excessive amount of the angiogenesis induced by the hypoxia induced factors. And on cut section we can see, in case of the smaller lesions there will be yellow tan lesions, small lesions, and these larger lesions seems to be hemorrhagic, necrotic, and cystic. Hemorrhagic, here we can appreciate various spots of the hemorrhage, and cyst-like formation. These are the various cyst-like appearance we can appreciate in the adrenal medulla once the lesions get enlarged. And next we see why the chromaffin name is given to the cells. It's because that these cells secrete the catecholamines, and if we inject potassium dichromate, then those tissues oxidize this potassium dichromate and give the dark brown coloration. And this happened only because of the presence of the stored catecholamines, and thus the term chromaffin is given. And next we see the microscopic features of the pheochromocytoma. First, we can appreciate that there is a well demarcated border, and various other features like sustentacular cells and gel valin nest-like patterns appears, and these are arranged in clusters or balls. We can see here they are arranged in clusters and balls, and these uh, clusters of the balls are surrounded with the sustentacular cells. So these cells are, we can say, appreciated such types of the cells, sustentacular cells, and delicate blood vessels surround this gel valin nest present in the pheochromocytomic tissues. And tumor cells, their shape could be polygonal or spindle shape. In the cytoplasm of the tumor cells may contain even dented, fine granular and red purple cytoplasm, and nuclei could be round, oval with prominent nucleoli. and there is a prominent nuclei and stippled, that could be stippled one, and that is present as the salt and pepper like nest, the sustentacular cells which is surrounding the nest, that are the small supportive cells, and this can be seen under the S100 protein immunostaining. And stroma has a delicate fibrovascular stroma, and that separates the nest of gel valley. This much for now. Thank you.